So yesterday we discussed some of the issues in Mendelian genetics. Okay. Total there are 4 laws are there. So total 4 laws are there. So first one, Loki. So Loki has 2 genes or 2 alleles. So this alleles terms were used by no. So Mendel used as a factors. So 2 factors as a unit responsible for the formation of a trait. So responsible for formation of a trait. So that is the first point. Now is it visible? I don't think so. So two alleles are a unit of factor. So unit of factors. So law of unit of factors responsible for the formation of a trait. So trait or else character. So in this unit, one is coming from the father, another one comes from the mother. So second point, if these two alleles are same, like AA. So on the chromosomal pair, both are AA. That means both are same factors. That we call as a homozygous allele. Homozygous allele. So homozygous Loki. So homozygous Loki means both alleles are same. So if both alleles are differ, like AO or AB, AO, AB or BO. So here different alleles are present on the same Loki. So in this context, we call as a heterozygous Loki. So heterozygous Loki. So now third one you can see. So law of dominance and recessive. So law of dominance and recessive. So when two contrasting factors, so two contrasting factors means different factors in heterozygous conditions. If they are comes from two parents, only one parent one expressed. So one parent one expressed. So in this case, A comes from one parent, O comes from another parent. And because they are two distinct alleles, only one allele expressed in the work. The remaining alleles are suppressed, silenced. That means those allele expressed we call as a dominant allele. Okay. So those alleles suppressed we call as a recessive allele. So that means, so genotype wise, it is present in an individual. Genotype was AB or AO. That means the individual contain both the alleles or both factors. But the problem is only one allele functions, another allele function is suppressed. So only one allele function is expressed, another allele function is suppressed. But theoretically, if we observe, the genes are present in an individual. So though in an individual, the genes are present, only one person, one parent gene is expressed. So only one parent genes are expressed, another parent genes are suppressed. So that we called as a heterozygous loci. So that we called as a dominant allele and a recessive allele. So dominant and recessive is a condition we always use to denote when heterozygous condition. In homozygous condition, because both are same, none of the allele is dominating, none of the allele is so recessive, both are same. So heterozygous condition comes only into the picture, dominant and recessive comes into the picture only this kind of heterozygous. So that means this is the core of the Mendel laws, core of the Mendel laws. And third law we have seen is like law of segregation. So law of segregation. So law of segregation means in a genotype. So in a genotype, while they are forming into a gametes, only one gamete receives one factor, another gamete receives another factor. So this we call as a law of segregation. That means, so in our pure biology, cell division time, during the metaphase, okay, metaphase 1. So the chromosomes are aligned as a pairs. Okay, chromosome aligned as a pairs, and each chromosome in pair, they go to the two sides, opposite sides, that later formed into a daughter cells. So this is talking about Mendel talking without knowing the biology, cells existence, chromosomes. He said that two factors are present in every character formation, trait formation. These two factors are shared between the two gametes, that he called as a law of segregation. Okay, so these are all the things yesterday we had seen by taking the example of garden pea. So garden pea plant, so where one is tall and another one is short. Okay, so these are all two contrasting characteristics. When two contrasting characteristics are present, in the next generation only one is expressed. That expressed one is called dominant. Okay, so suppressed one is called recessive. So that means, so these are all the things over a period of time the geneticists as well as medical practitioner documented. So documented things. So in garden pea plant, Mendel was documented. In later medicine, see, so we know that if a person has A and O genotype, okay, that genotype is, that means O, only A is expressed, and A is expressed, O is silenced. That's why A called as a dominant. Okay, so in an individual, when they applied the antigen, antibody reaction, only A blood group appeared. So O is suppressed. That means they come to know that A allele and O allele act as a law of, they inherent according to the law of dominance and recessive. That is because he had already given a set of laws that they are applied in the real time. When two contrasting characteristics are present, so whatever expressed, and they listed that expressed trait as a dominant. Okay, nobody knows which one is dominant. When they fertilized and the appeared character only, they recognized it is a dominant because it follows the law. And again in the next generation O appeared, they come to know that Mendel laws are always perfect. So now, so we will take the human examples. So human examples. So PTC substance is there. So PTC substance, it's like one of the chemical component. So it is not harmful. So phenyl, so thiocarbamide. So phenyl thiocarbamide. So this is the substance. It is a chemical substance, but not harmful. So these substance, humans are classified into tasters and non-tasters. So some of the human beings reported this substance as a bitter in taste. So bitter in taste. And other people, they reported it has no taste. So nothing, they felt like any taste sensation. That means no taste. Okay, no taste or non-tasters. So that's why we can say these are all the two contrasting characteristics with related to the PTC substance. So these kind of people are 25% human beings can able to taste the PTC substance, and these are all nearly 75% people are there. So in a case when a taster person married non-taster person, so what kind of children we will expect? Okay, so now if you know that taster is dominant, then answer is very easy. All children are born with a taster. Understand? So for suppose you can take taster as a capital T. So opposite, right? Non-taster, that we will write a small t character. Okay. So parental generation, if these two got married, so his genotype is like a TT. So parental generation. So for suppose if we think that non-taster is a female, small t. Okay, taster, non-taster. So what about that gametes formation? Gametogenesis, gametes formation, capital T, capital T. Okay, what about that small t, small t? So this is the parental gametes. So parental gametes. So these parental gametes, they apply random fertilization. So random fertilization means every gamete has equal probability of mating with another person's gamete. So this is mated with any one of this one. So what is the resultant one is capital T, small t, and capital T, small t, and capital T, small t, and capital T, small t. So this is all about all the progenies are. So this is nothing but capital T, small t. So all the progenies are heterozygous. So heterozygous tasters. So this person also taster, right? This person we called as a homozygous taster. So homozygous taster. Homozygous taster. So this person is called heterozygous taster. Why? Because only T is expressing, small t is silenced. That means here, due to law of dominance and recessive, due to law of dominance of taster ability, due to dominance of taster ability, all the progenies born from a homozygous taster, non-homozygous taster, all the children are born with tasting ability. So taster, understand? This is also taster. So this is the combination we come to know that. Okay, so this can be applied for any kind of trait, so which has the property of dominance and recessive. So if you take the so diabetes, so diabetes is a inheritance disease, but diabetes is a recessive gene. Okay, so human beings are non-diabetic. So non-diabetic, diabetic, two type of populations are possible. So diabetic. So non-diabetes, I am taking as a capital D. Okay, small diabetic, I am taking as a small d. If a non-diabetic person, that is capital D, homozygous non-diabetic. So homozygous non-diabetic. So homozygous diabetic. Homozygous diabetic. So if they both get married, then what would be the result? So this is the case 2 I am explaining. So two gametes are possible. So this is called F1 generation. So these are all the individuals are F1 generation. It's not yet over. There is continuation of the discussion. So here you can see capital D, capital D. So it is called gametes formation. Gametes. So here small d, small d. That means the individual who possessed, I'm telling that. So next, random fertilization. So here capital D, small d, capital D, small d, capital D, small d, capital D, small d. So four individuals are possible. In this four individual, heterozygous. This is heterozygous condition. Heterozygous condition. Heterozygous non-diabetic. So heterozygous non-diabetic. So everyone is a non-diabetic, 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 non-diabetic. But still he has the character of, still he has the gene of diabetic gene. So small d is a diabetic gene. Understand? So small d is a diabetic gene, capital D is a non-diabetic gene. But here I said that means non-diabetic, diabetic, both are contrasting factors, contrasting alleles. In contrasting case, only one is expressed. Expressed we can call as a dominant one. Recessive we can call as a non-expressed one we call as a recessive. In this case, capital D is expressed, small d is non-expressed. That means capital D. So here small d is suppressed means diabetes is a recessive disease. Diabetes is a recessive disease. So whenever a person has diabetic gene and another gene is non-diabetic, he cannot express the symptoms. Here, so taster is a dominant gene. Tasting ability is a dominant character. Diabetes symptoms appearance is a recessive character. So in this case, all the individuals, though they have the diabetic gene, but externally, physically, they never exhibit any diabetic character. Understand? So this is how, if any one of the parent is diabetic, still their parents, their children are non-diabetic. But that doesn't mean the story is not yet over. Okay, so they produce like 4 children. They produced 4 children. Say one of the first one is we can presume as a girl. First one we can presume as a girl. Okay, she is the female. Okay, she has non-diabetic. Non-diabetic. And what is the genotype? Capital D, small d. Non-diabetic, capital D, small d. From another family, boy. From another family, boy. Because these are all siblings. They cannot marry among them. So we can take the same kind of family another, in which we can choose heterozygous non-diabetes. Okay, the boy is non-diabetic. So non-diabetic, he has the character of heterozygous non-diabetic. So capital D, small d. So now if they go for the fertilization, they got married, they produce the children. They produce the children, and these are all gametes. Capital D, small d. So these are all the boy gametes. Capital D, small d. So now you can see after in F1 generation, or this is F2 generation. So F1 generation, from 2 families who are taken and they got married. In F2 generation, capital D possible, capital D, small d possible, capital D, small d possible, capital D, small d is possible. So how many types of genotypes appeared in the F2 generation? 3 types of genotypes. So 3 types of genotypes. That means the individual children born with 3 types of genotypes. Capital D, 100% non-diabetic. Non-diabetic. Okay. So capital D, small d, heterozygous non-diabetic. Heterozygous non-diabetic. So non-diabetic. So small d, small d. So because there is no other gene to suppress this diabetes gene, both are diabetes gene. So then this person is a diabetic person. So diabetic person. So this is how in F2 generation, again the diabetes appeared. In grandmother, diabetes is there. In grandparental generation, one of the person is diabetic and another person is non-diabetic. So in the progeny, in an offspring generation, in F1 generation, nobody has diabetes. Okay. In F2 generation, it suddenly reappeared. So this is the medical application. You can see, so in a diagnosis process, if any one of the individual has diagnosed as diabetes, and they will ask like, is there any of your parent having diabetes? Okay, so in that case, if both the parents have the diabetes, then it is derived from the parental. If both parents are diabetes, okay, and then it is the possibility of deriving. If both are heterozygous diabetes, there is a possibility. If both parents are heterozygous diabetes, means in grandparental generation, if any one of the person has diabetes, understand? So that means here in one of the generation, the gene is present but characters absent. In F2 generation, the characters are appeared because of recessive condition. So why? So recessive. So homozygous, here there is no dominating gene. It became a characters appeared, affected person. Affected person. What about this person? This person is non-diabetes but heterozygous. This person is a carrier of the carrier affected person, means if a person has the gene of a particular disorder or disease, but if he never expressed any symptoms, that person we call as a carrier. If a person has the disease genes and the person expressed the symptoms, that person we call as a affected person. Here that person has insulin deficiency. Here the person never had insulin deficiency, still he has the genes of diabetes causing. Here this person has nothing. This person has no genes related to the diabetes. Understand? So this is how we can apply in the human population. So see here. So genetic disorders generally present as a recessive. So human populations have too much of genetic disorders. Too much of genetic disorders. And you, me, and everybody has the genetic disorders. So genetic disorders. But most of the human genetic disorders are recessive in character. So recessive in nature. So that means I have so many genetic disease causing genes, but it was suppressed by normal genes. So normal genes. So that's why any genetic disease which is masked as a carrier, for that reason, which is never expressed in the individual, so that the person is healthy, looking like healthy. So if more persons are expressing these recessive genes, means the population must have heterozygotes more in number. So conclusions, I can write some conclusion you should understand. So that means generally human population has large number of genetic disorders. So disorder causing genes are present, but they are not affected. The reason means when they affect, when the homozygous condition only, the disease's nature is nothing but a recessive. Recessive only expressed in the homozygous condition, but recessive genes are always suppressed by a normal gene. That means in a population, so conclusion you can see, in a population, more recessive genes are present. So more recessive genes are present. Okay. So, but few people are affected. Few people are affected. So few people are affected means here more recessive in every human population, more recessive genes are there, but the persons are affected persons are very few. In that case, what would happen? Most of the human populations are heterozygotes. Heterozygotes, not the homozygous. So if it is the more number of people are homozygous, then what would happen? If more number of people are homozygous, might be 50% of people are like DD, 50% of people are DD, 50% of people are small d. In this case, in every population, half of the population must have affected. But very few, 10%, 5% people are affecting, means that means 95% of people are non-affected. That is capital D, small d combination. So this is how the relevance of, in medical field, the relevance of dominance and recessive is. So humans have most of the recessive genes. That recessive genes are always suppressed through heterozygotic condition. These heterozygotic conditions are prevailed when random mating occurred. So random mating occurred. So for suppose I'm telling like this. So if a person has very simple, so practical application is there. So you take a person. So this family one, okay. So family one has one parent diabetic. Okay, another parent is normal. They produce the children. Child one, child two. Okay, 4 children are there. In another family, okay. So one is diabetes. Okay, another one is normal. So they produce the 4 children. So 4 children. So means here diabetes means small d, capital D, small d, capital D. So here all are capital D, small d, capital D, small d, capital D, small d. Okay. So capital D, small d. So if they enter the matrilineal alliance, okay, here 2 are female sisters, 2 are brothers. Here 2 are brothers, 2 are sisters. They exchange the mating. So exchange marriages. In exchange marriages, so these 2 are married, these 2 persons. These 2 are married, these 2 persons. So now every couple produces diabetic children. So every couple produces diabetic children. So now how many couples are there? So now couples are 4. Here 2 males are there. 2 males, their children's. So because patriarchy, females of this group, this family moves here. Females of this family moves here. So 2 sons of this family with their wives and their children. So here, so same 2 brothers and their children. So in these children, some of the people are affected. Okay, two persons are affected. Two persons are affected with diabetes. So remaining children, so because they comes under the cross cousins. So this group again marry into this group. This group is again married into this group. So these children are again married this. These are married again this. Now the children, the number of diabetic people increases. Understand? So in the next generation, again, this group is married to that group is married to. That means over a generations increasing, the genes are exchanged between these people. The recessive became homozygous. So recessives became homozygous. So more homozygous increases and less heterozygotes come into being. So less heterozygous. The 2 groups. So this group is growing bigger size. This group is growing bigger size because both are exchanged their children. The group increases, but the genes are exchanged between the 2 groups forever. So in these groups, number of heterozygotes decline. So number of heterozygotes decline, but homozygous increase. So homozygous increase. So homozygous increase means it is dangerable because homozygous there is a condition called diabetes. Understand? So random mating means, so when they observe that some of the children born with diabetes, if they give their children with another families rather than the other family, if they choose in the unrelated family, so they have healthy genes, non-diabetic genes are there. That genes come here and they start dominating the diabetic genes. So that means always marriages should happen outside the, if closed circuits, the marriages are happening generation after generation. So heterozygotes decline, homozygous increase. So generally human population have more recessive genes. So homozygous conditions are easy to get the recessive genes expressed, or recessive genes, the people are affected. So if they marry unrelated persons, the normal genes are entered and they dominate over the recessive genes, so that every individual looks like a normal and healthy. So according to genetics, the marriages So marriages among closed individuals among marriages among closed individuals over a generations. So over a generations result into expression of the recessive genes. So expression of the recessive genes because more homozygous. So more homozygous. So this was observed by the human beings, many generations back, like 2 lakh years ago, 3 lakh years ago, to prevent this kind of genetic disorders. Since the beginning of the human race, we have this recessive genes. To prevent this one, they created the marriage rules like exogamy. Exogamy. That means a person born in a one group. Here males and females are born in this group. So they should not marry among them because they have the common ancestors. They should always look out the another groups to get the marriage. Understand? So this is how Mendelian laws are practically important. So the cultural rules. Some exogamy prevent the homozygous. Homozygous it prevent, heterozygous it increases. So here exogamy also referred as a random mating. So in every generation the mating should be unrelated individual, not the related individual. But endogamy operates. So endogamy operates like you can take that cross cousin marriages, parallel cousin marriages. Okay, cross cousin marriages, parallel cousins, cross cousins, uncle niece marriages. So these are all generally very high in the southern India. So whenever brother sister marriages, it was prohibited all over the world, brother sister marriages. So these are all comes under close individuals. So if the matings are more, marriages more among these people, their children are suffer with genetic disorders. So why genetic disorder? Their children are born with homozygous conditions. Born with homozygous condition and they are affected because recessive. They are affected because of recessive. So this is the practical application. So how cultural rules are responsible for increasing the genetic disorders, or what kind of cultural rules are responsible for to maintain the less number of genetic disorders in a population. So exogamy reduces the genetic disorders. So the cross culture preferential marriage rules are there. Parallel cousin, cross cousin, uncle niece marriages, brother sister marriages. These are all increasing the genetic disorders in the population. So that's why most of the societies they prohibited. Wherever the society is practicing, these are all primitive societies. So no modern society allow these marriages. Only primitive tribal societies have. So modern civilized people, they never marry cross cousins, parallel cousins, uncle niece marriages. So the reasons are this. So culture can be increased the genetic disorders. Culture can be reduced the genetic disorders. Understand? So now we are going to the somewhat superior concept. So here, so much of thing is there. So this is how. So why recessive genes are important? Why dominant genes are important? Most of the cases, majority dominant traits are useful for the humanity. Okay, majority cases, most of the recessives are diseases or disorders. So dominant alleles are helpful for the survival of the organism. Recessive genes they come due to mutations. They come due to mutations. Mutations are generally negative in nature. Understand? So negative in nature, they are negative consequences for the life of the individual organism. So this is how. So now we are going to the frequency wise. So now we are going to the frequency wise. So we will apply. So roller and taster, human examples, taster and non-taster. So tasters are two types. One is capital T, homozygous taster. Another one is capital T, small t, heterozygous taster. So non-taster is only one case, small t. Because it is a recessive, the ability only comes if there is no dominating gene. Okay, so that means tasting ability is a, so the character here is, character or trait here is, so character or trait here is ability to taste. So ability to taste, PTC. So this is the character. So character or trait name is ability to taste the PTC. So how many alleles are present? Capital T, small t. Two genes are there. So if an individual has capital T, homozygous taster. Okay. So if the individual has small t, non-taster. So here we don't need to write homozygous non-taster. Why? Because there is no heterozygous non-tasters. So if these two are present, so in human population it can be TT or small t. So because of this mating, capital T, small t come. So this combination we call as a heterozygous taster. Heterozygous taster. So in a chromosome, in a chromosomal loci, if only one allele is present, here alleles are, what are all alleles? Capital T, small t. So because human population has the two alleles, so it is spread among the human beings. Human beings has three type of genotypes: capital T, small t, capital T, small t, small t. So if only one allele is there, like capital T only present, so every human being is a capital T, capital T, capital T, capital T. Understand? That means this condition we call as a human beings are polymorphic. Polymorphic. When in a loci, if more than two alleles are present, if more than one allele is present, so that results into polymorphic population with the ability of taste of PTC. Humans classified as a homozygous taster, non-tasters, heterozygous tasters. If three alleles are present in the same chromosome, same loci, then it becomes six. Six type of human population possible. If four alleles are present, five alleles are present, if many mutations are there, many alleles coming in the same loci, then human population diversity increases. If more alleles in a loci, more diversity among the human being. Understand? So the diversity increases. So diversity increases with more alleles in a loci. With more alleles in a loci. So now you take chromosome 9th pair. Chromosome 9th pair. So one of the loci is ABO blood group. How many alleles are there for ABO blood group? A allele, B allele, O allele. Here. So this combination, we can write AA people. Okay. We can write AO people. We can write AB people. Three combination. Now fix that BB people, BO people. Okay. So BA already over. So next OO people. So if three alleles are present, human population diversity increases to one genotype, second, third, four, five, six. If four alleles are there, diversity further increases. In another loci, if four alleles are there, diversity increases. If two alleles are there, three type of genotypes. So genotypes are three. So for tasting ability, how many genotypes are there? Capital T, small t, capital T, capital T, small t, small t. Three genotypes. How many phenotypes are there? Output. Phenotypes are two only. So genotypes are three. Phenotypes are two only. So this person is a taster. This person is a taster. This person is non-taster. So that means human populations are phenotypically two, taster and non-tasters, but genotypically three. Understand? So phenotypes and genotypes. Output, total output is, even it is heterozygous, but output is taster only. He is not a non-taster. But this is homozygous, but output is taster only. That means phenotypically population is categorized into taster and non-taster. Genotypically, three type of genotypes are possible. Understand? So how many alleles are there? Alleles, only two. So when a loci has two alleles, three genotypes, okay, two phenotypes, four offsprings. That is the frequency. So because we are looking at the single character with contrasting alleles. So ability to taste PTC is a single character or a single trait. But with the contrasting alleles, capital T, small t is the contrasting alleles. When a character has contrasting alleles are 2, in this situation, alleles numbers are 2, genotypes are 3, phenotypes are 2, offsprings are 4. So offsprings are 4. Offsprings are 4. So this is the frequency of the single trait. So frequency of the single trait. So now I am introducing second case. So we will measure generally frequency of inheritance. So frequencies of inheritance. First one, number of alleles. Number of contrasting alleles in a loci, that is n. Okay. So number of gametes. Number of gametes 2 power n. Okay, number of gametes and phenotypes. So number of gametes and phenotypes 2 power n. So number of alleles. Here number of alleles. Okay, I will look at. So number of genotypes 3 power n. So number of offsprings. So number of offsprings 4 power n. 4 power n. So number of alleles or number of characteristics we can fix. So number of characteristics. So number of alleles and number of characteristics. Whenever there is a contrasting characteristics, we will take. So tasting of PTC is a one character. Okay. So if you want, I will explain one more time. Okay. So you need further elaboration. We will see the next case. Rollers and non-rollers. So human beings are classified into rollers and non-rollers. So what do you mean by rollers and non-rollers? So rollers are nothing but some people able to twist, rolling their tongue. Like this, some people able to roll their tongue. Majority people not able to roll their tongue. So not only the ability of tasting the substance, when it comes to tongue rolling also, some people able to do that we call as a rollers. Some other people called as a non-rollers. The same process. So roller we call as a capital R. So opposite we should take as a small r. Gametes formation. Okay, small r. So the rest of the story, capital R, small r. Okay, 4 genotypes. Okay, so next, each one is R is a dominant. Roller is a dominant character. Roller, dominant. That means capital R is dominant. So non-roller, small r. So small r causes non-rolling ability. So it is recessive. That means all the individuals are able to roll their tongue. So roller, heterozygous roller. Understand? So now this is one family. From another family, if we take the non-roller, okay, roller only, but family 2, he has this one, heterozygous roller. So they both got married. So these two got married. So she is from another family, but heterozygous roller. Okay, so this is the heterozygous roller from the family 1. This is family 2. They both got married. They produce again they produce. Okay, again the story you can see. So I will explain here itself. So this and random fertilization. So capital R has the possibility of mating with any gamete. Capital R, small r. small r, capital R, small r, small r. So now this is roller. This is roller. So these two also roller, but heterozygous. Heterozygous. Non-roller. Non-roller. So these are gametes. So these are gametes. These are offsprings. Okay, these are all offsprings. So if you go to the top also, so these are all parents, parental generation. Parental generation. Parents. Okay, so their gametes. So their gametes. So these are all the F1 generation. Okay, these are all offspring. So in F1, how many children born here? So number of characters. See, n is equal to number of characters. One, what character we have taken? So if an individual has roller, taster, and how this roller and taster person married a non-roller, non-taster. A single person might have roller and taster, and another person might have non-roller and taster, non-roller and non-taster. In this combination, number of character inherited by the offsprings are we are studying for 2. That means n is 2. But in this case, we never bothered about the tasting ability. Only roller's case we observed. That means number of characteristics observed in the inheritance process is only 1. But here number of characteristics are observed in 2. This is difactorial. We can say single factor. This is called two-factor inheritance. Two factors inheritance or two contrasting factors inheritance. That is different case. Okay. In this case, the character is only 1. That is whether a person is roller or non-roller. So that is the only one case. Tongue rolling ability. Character name is tongue rolling ability. Tongue rolling ability. So this is the character. So character is 1. Okay. So number of alleles. How many number of alleles are there? Capital R, small r. So alleles are 2. So you can place here equation. Number of characteristics are 1. N is equal to 1. So number of gametes or number of alleles. So alleles we cannot identify. Alleles also we can identify. 2 power n. Alleles also 2 power n. So now you can see how many alleles are present here. So 2 power n is equal to 1. 2 alleles are there. How many gametes are formed here? So in parental generation, capital R, small r, capital R, small r, small r. 2 gametes only. One is capital R gamete, another one is small r gamete. So that means gametes are 2 power 1, 2 power 1. That is 2 gametes. So gametes formula is you have to substitute n is equal to 1. So gametes are 2 power 1. That is 2. These are all the alleles. These are all the gametes. 2 gametes only possible. So if you want, you can see here in F2 generation also, F1 people, they too produced only 2 type of gametes, capital R, small r gametes. Understand? So now you can see phenotypes. How many phenotypes are possible in every generation? Here roller and non-roller. Here rollers are there. In the F2 generation over, what type of phenotypes are there? Roller, non-roller. Okay. So 2 phenotypes are there. 2 power 1. So 2 power 1. That is roller and non-roller. In F2 generation ending, we will take the characteristics. The frequency measured in the F2 level. So F1 level is nothing but a half of the inheritance. When F2 completed, complete inheritance chart is prepared. So F2 level values we will take. So in F2, rollers coming being, non-rollers coming to being. But what is the ratio? So two are there, but rollers are 3 and non-rollers are 1. So you can see How many rollers are there? This is the roller. This two also roller and final. That means 75 is to 25 or 3 is to 1. So 3 is to 1 phenotypes are there. Rollers and non-rollers are 3 is to 1 ratio. Two phenotypes are there, roller and non-roll, and their frequency is 3 is to 1. So now you take number of genotypes. So number of genotypes are 3 power 1. That is 3. What are all the genotypes? So roller, 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 non-roller, roller, roller. So 3 types of genotypes are there. So number of offspring, 4 power 1. So that is 4. So what are all these 4? So this is 1, second, third, 4. 4 offsprings are there. So when the character is 1, alleles are 2, offsprings are 4, phenotypes are 2, genotypes are 3. Okay, so here, so genotype frequency. What is the genotype frequency here? So what is the genotypic frequency? These are all the genotypes, right? 3 genotypes are there. Genotypic frequency are capital R 1, capital R small r 2, small r is 1. Understand? So 3 genotypes are there. 3 genotypes are there. Capital R 1, capital R small r 2, and small r 1. 1 is to 2 is to 1. Understand? So here the ratios, when single character with contrasting alleles are present, so it has the genotypic ratio. Genotypic ratio is, so genotypic ratio is 1 is to 2 is to 1. Okay. Phenotypic ratio is 3 is to 1. So 3 is roller. Okay. 1 is non-roller. So 1 is capital R, 2 are capital R, small r. So this is the formula. So this formula you will get better understand when we taken two traits with contrasting alleles. So now I am introducing the another individual inheritance. So another individual inheritance. So two persons got married. So two factors are difactorial inheritance. So till now we have seen single factor inheritance. If a person is taster and another person is non-taster, so these two are related to tasting ability. Okay, another couple we have seen that inheritance within this couple is whether rollers and non-roller, how it is passed across the generation in another couple. So now in a couple, if both characters are present, how these are passed to the offspring. So that we are going to see. So now we will take two individuals, male and female. So because they only get married, male is a taster roller. So till now, if male is a taster, female is a non-taster, how it is going to be inherited in the offspring and their frequencies we observe. So now two characters in an individual we are observing. Next 3 characters, next 4 characters. So this is how unlimited characters in an individual we can observe. Okay, if we know one character, then second character, third character. This is how multiple characters, because every individual has multiple characters. So to simplify that, I started with a single and I will conclude with the 2 and we will go for the final conclusion. So taster and non-taster, taster and roller. Okay, they are the parents. Female is non-taster, roller, non-roller. So for simplification process, in one of the, so every human being has 23 pairs of chromosomes. For suppose the tasting related loci is present somewhere else. So TT. Roller loci is present somewhere else in another pair. So RR. Because two locis are not present on the same chromosome, they present in the total chromosomes, but different pairs they present. Okay, so this is why capital T, capillary. So now for simplification process, this is the inside the chromosomes. But simplification process, taster means capital T. Roller means capillary. So non-taster means same kind of thing. So small t. Okay, roller means smaller. So this is the genotype of the father. This is the genotype of the mother. Okay, now gametes. Earlier it is easy. Capital T, small t is present. Capital T is present. So now they have to produce the gametes. How many gametes are possible here? How many gametes are possible here? So gametes formation. Generally two gametes come. Understand? So from this, generally two gametes only come. So if this chromosome, capital T comes here, this T comes to here. Understand? So here capillary comes here, one capillary comes here, another capillary goes to this one. That's it. Okay. So TR, one gamete. So here TR, another gamete. So if you want very small, okay. So you can take the two chromosomes, two loci are one. So one is capillary, capital T. But if you want complex calculation, it is difficult. So gametes, how it is formed? This goes to one gamete, this goes to another gamete. So TR. But in genetics, in reality, it is not that much simple. So this is how TR is the complex, gametes. So here two gametes, small t, small r. One loci related to tasting ability, small t. Another loci related to rolling ability. So two gametes. So this chromosome goes to one side. This chromosome goes to another side. TR. It's not that much simple. When three characters are coming, very difficult. So now, so this is the individual. Now they are going to the fertilize, random fertilize, gametes. So gametes and segregation. Law of segregation. Okay. So this is gametes and law of segregation. So now gametes form on random fertilization. If this one is fertilized with this, so this one is fertilized with this, and this one is fertilized with this, and this one is fertilized with this. Four combinations are there. Capital T, capillary, small t, small r. Okay. So capital T, small t, capillary, small r. Same here also. Capital T, small t, capital R, small r. So this one, capital T, small t, capital R, small r. Capital T, small t, capital R, small r. So now you can see, so this person is a taster roller. This person is a non-taster, non-roller. So they got married. What are all the offspring phenotypes? So because children are born, F1 generation came into being. F1 means filial. Filial means offsprings. F1 generation children. So this is heterozygote. So you can see tasting ability. If both are capital T, homozygous taster. Okay, both are capital T, small t, heterozygous. So heterozygous taster. What about roller? Heterozygous roller. So that heterozygous taster roller. Same taster roller. Heterozygous taster roller. So heterozygous taster roller. All the children can able to taste the PTC substance and able to roll the tongue. So now this one of the family married another family. Okay, this person married the family two. Family two. So family two, we have to take same kind of thing. Capital T, small t, capital R, small r. So heterozygous taster roller. Heterozygous taster roller. So now they produce gametes. They produce gametes. So what are all the gametes here? How many gametes are possible here? So how many gametes are possible? See, taster, non-taster. Taster, roller combination. Taster, non-roller. So taster, non-taster, roller. Non-taster, non-roller. So here how many gametes possible? Not two, 4 gametes. Okay, so that's I will explain. So here it is capital T, roller means capital R, so non-roller means small r, roller means capital R, small r. Okay, so small t. Okay, so capital T, capital R. Okay, so capital T, small r, capital T, small r. So small t, capital R, small t, small r. Four gametes are possible from here. TR, capital TR, capital T, small r, small t, capital R, small t, small r. So here also 4 possible. Same gametes. Capital TR, capital T, small r, small t, capital R, small t, small r. So this is 2 persons. So now this one has random mating with 4. This one has random mating, another 4. This one has random mating, another 4. This one has random mating, another 4. Total 16 offsprings coming to being. So in F2 generation, if 2 characteristics we consider in an individual in a family inheritance, total number 16 offsprings are coming to being. So this we cannot write here. That I will explain further. So first you can tell me why 4, why 2 here. So here also 2 characters. Okay, taster, non-taster. Okay, taster roller. In a parental generation, so taster roller. But there I wrote only 2. But here I wrote 4 gametes. So why? So that is the reason. For suppose first pair, second pair, third pair, fourth pair, somewhere else like 8th pair, 9th pair. So this is how 23rd pair. So this is how in one of the pair rolling genes are present. Okay, so rolling genes are present. In another pair, so there is like tasting genes are present. Okay, so now this has to be formed into a gametes, 2 gametes. So 2 gametes should come from this combination. Gametes has to come. First of all, in one of the in one of the gamete, this chromosome come, that is capital R. Okay, so then automatically small r goes to the another. Okay, so now here these are all the homologous chromosomes. Pair means homologous chromosomes. So these are all gametes formation. Means when do gametes occur? Meiosis process. In meiosis time, metaphase 1 crossing over process involves. Crossing over means homologous chromosomes exchange their genes. So for suppose these two regions were exchanged, this was given, this one is given, it is not participated. Then what happens? In the first one, capital T only present. In the second one, small t only present. So now capital T. Okay, now this is the small t. If crossing over involved and this below, it's not the below portion and above portion got affected, then what will happen? Switching occurs. This side is small t comes, this side capital T comes. And in this combination, capital R and first chromosome, capital R and small t. Okay, so second one, small r and capital T. Small r and capital T. So how many possible? Because of crossing over. So in the earlier case, both are same. Both are here capital R, capital T, capital T. If both are capital R, capital T, capital T. If crossing over involved, not involved, nothing will change. Both genes are same. So if crossing over involved, on the first capital T present, second on capital T only present. Here also first one capital R, second one capital R. But contrasting genes. Okay, if first one is capital T before crossing over, after crossing over gametes form. In this case, capital T plus small t comes. So in one case, the genes whether involved in the crossing over, this one RT and RT before crossing over. If the genes place crossing over involved, these two also occur. So this is how 4 gametes are possible in 2 characteristics inheritance. Understand? So now, so this 4 we have to see one by one. Okay. So Punnett square, we call it as a Punnett square. So Punnett square. So Punnett square means 4 gametes we will take. 4 gametes. So what are all these gametes? Capital T, capital R. One gamete. Capital T, small r. Third one, small t, capital R. Small t, small r. 4 gametes. So here capital T, capital R, one gamete. Capital T, small r, another gamete. Small t, capital R, another gamete. Small t, small r. So this is 16. So why 16 comes? You can see here. So this one has random mating. Capital TR can mate with any 4. So small tr can mate with 4. So small t, capital R mate with 4. Small t, small r mate with 4. So now we can see. So this I write here. Capital TR can mate with 4. Capital T, small r can mate with 4. Small t, capital R mate with 4. Small t, small r. So this is how Punnett score is better. If 3 contrasting character, then it is 2 power 3. 8 gametes will come. If 2 character, 4 gametes. 3 characters, 8 gametes. Then Punnett score increasing bigger. That we cannot discuss. Okay, very bigger one. So here you can see. So this combination, capital T, capital R, capital R. So this combination, capital T, capital R, small r. This combination, capital T, small t, capital R, capital R. This combination, capital T, small t, capital R, small r. So 4 combinations, 16 offsprings. 16 offsprings in F2 generation. So here, capital T, capital R, small r. Capital T, small r. So this one. Capital T, small r, capital R. So this one and this, capital T, small r, small r. Okay. So this one and this, capital T, small t, capital R, capital R. So capital T, small t, small t, capital T, capital R, small r. So now you can see third one, small t, capital R, capital R. So this you can see small t, capital R, small r. So now you can take this gamete has the four probabilities, capital T, capital R, small r. This one, capital T, small r, small r. So this one, small t, okay. So capital R, small r. So last one, small t, small r, small r. So this is the 16 offsprings, 16 gametes. 16 gametes. So 16, not 16 gametes, 16 genotypes. 16 offsprings have 16 genotypes. So each one is a genotype because two characteristics we had considered, four letters we are showing. So each character 2 letters. 2 character means 4 letters. 3 character means 6 letters. This is how two letters indicates one loci. So now you can see capital T, capital R. That person is a pure taster and roller. So taster, roller. Okay. So capital T. That means for tasting ability, taster. So capital R, small r means capital R dominates. That means this is the rollers. This is the roller. So what about this one? Taster, roller. This is taster, roller. Okay. So now you can see taster, roller. So this capital T, small r, taster, non-roller. This child born with taster, non-roller. This is child with taster, roller. So this is child with taster, non-roller. Okay. So now next child is taster, roller. Taster, roller. So non-taster, roller. Non-taster and roller child. So non-taster and roller child. And if you go to the here, taster, roller. So taster, non-roller. So non-taster, roller. Non-taster and roller. So non-taster, non-roller. Non-taster, non-roller. So this is the 16 gametes, 16 offsprings and a 16 offsprings. And what are all the phenotypes here? So phenotypes are taster, roller. Okay, non-taster, non-roller. So first one taster, roller, last one non-taster, non-roller. In between taster, non-roller, taster, non-roller, non-taster and roller. So these are all the phenotypes. So genotypes are 16, phenotypes are 4. So this ratio is taster, roller. So taster, roller are 1, taster, roller 2, taster, roller 3, 4, okay, 5, 6, 7, 8, 9. 9 people are taster roller. Okay, so the remaining taster non-roller. So taster, this one is 1, non-taster, non-roller is only 1. Okay, so taster non-roller is 3. So this is the frequency. So now I will show you. So I will show you some chart. So only one character. So only one character we observed among human beings, either roller or taster inheritance, not the both, either roller or taster. After F2 generation, So how many phenotypes are there? Phenotypes. 2 in number, that is taster and non-taster. So what is the ratio? 4:3 or 3:1. The ratio is taster are 3, non-taster is 1. So what are all the gametes ratio? Not gametes, genotypes. So 1:2, 2:1. So what are all those? Capital T, capital T, capital T, small t, small t, small t. This is after F2 generation. Phenotypic ratio, genotypic ratio. That means total offsprings, total 4, 1 plus 2 plus 1, 4 offsprings. Okay, so this is the phenotypes, genotypes. Okay, gametes only 2, character only 1. So n is 1. So when character is only 1, either roller or taster, what is the gametes? 2. So that is capital T or small t. Only 2 gametes are possible. Okay. So character is only 1. Offsprings F2 generation later 4. So genotypes are capital T, small t, capital T, capital T, capital T, small t, this one. So phenotypes are 2. Taster, non-taster only comes. Either the person born with a taster or non-taster. So now 2 characteristics. So now 2 characteristics, taster and roller. Both abilities we are observing. So that means number of characters, n is 2. Characters are 2. So now number of gametes. So number of gametes are 2 power n. N means 2. 2 power 2, 4. 4 gametes are there. So what are those 4 gametes? Capital T, capital R, capital T, small t, small r. Okay, so small t, capital R, small t, small r. 4 gametes are there. So 4 gametes. So next, offsprings, F2 generation later, offsprings 16. 16 means 2 power 4. So 4 power n, 4 power n. So 4 power n is the 16. 4 power 2 is equal to 16. So this is the total gametes. So this is the total gametes. In 2-factor inheritance, this is the 16 gametes. Okay. 16 gametes. That is 16 offsprings. Next, genotypes. So genotypes formula is 2 power 2. 2 power 3 power n. Genotypes are 3 power n. 3 power n means 3 power 2. 9 genotypes are possible. 9 genotypes are possible. So what are all these genotypes? So these are all genotype. So this is one genotype and this is another genotype. So this is another genotype. So if you go this one, you will get 9 type of genotypes, 3 power n. So this is one genotype. Okay. So this is capital T, capital T, capital R, capital R. So this is another one, two types of genotypes, capital T, capital R, capital R, small r. This combination again appears here. That means these two we can consider as a one type of genotype. So this is one, these two are, so overall this is three type of genotype. Then if you go to this one appear another, so four genotype. This is how 9 type of genotypes are possible. Okay, so 9 type of genotypes are possible. 3 power n. Genotypes is nothing but 3 power n. So n is 2, that is 9 genotypes. So what are finally phenotypes? So phenotypes are 2 power n, that is 2 power 2, 4 genotypes. What are all these four phenotypes? Taster, roller. Okay, taster, roller, taster, non-roller, non-taster, roller, non-taster, non-roller. So these are all the 4 genotypes. 4 genotypes comes from the 9 genotypes. 4 phenotypes. 4 phenotypes are comes from 9 genotypes. So this together produces 16 offsprings, and it is caused by 4 gametes. Okay, 4 gametes are result of 2 characters. Understand? So now, so another character you can take in the human beings. Okay, diabetes we can take. Non-diabetic is a dominant. Okay, diabetic is a non-diabetic is a dominant. Diabetes is a recessive. So if you take the third character in another case, so number of characters are 3. Number of characters are 3. Number of characters are 3. What are all those? A person is taster, roller, non-diabetic. Another person is married. Another person non-taster, non-roller, diabetic. So this is the third character. So if I take 4, if I take 5, tri, penta, hexa, this is our number of characters. Di characters, tri characters, tetra, penta. This is our number of character wise. So, but if you go this one, what are all the number of characters? So number of characters are 3. That is n value. So number of phenotypes, number of alleles also. Alleles if you take 2 power n. So 8 alleles. 8 alleles. Okay, 8 alleles. Alleles we cannot write. It's not required. So number of gametes. Number of gametes 2 power 3. That is 8 gametes. So number of phenotypes. Number of phenotypes 2 power 3. That is 8. Okay. So number of genotypes 3 power n. 3 power n means 3. 9 genotypes. 27. 27 genotypes. Number of offsprings. Number of offsprings. So 4 power n. 4 power 3. 16 into 64 offsprings. 64 offspring. If number of total characters are 3. If 4, again these values. So that's why the formula is there. Okay. The formula is there. So number of gametes, this you can omit. N is equal to number of character. So number of character in inheritance. Number of characters in inheritance. So that is the value is n. When number of characters is n, the total number of gametes formed are 2 power n. Okay. So the number of phenotypes are 2 power n. Number of genotypes are 3 power n. Number of offsprings are 4 power n. So by this formula, we can predict how many children are possible. Okay, how many children are possible? Understand? So we can predict or not by going this one. What are the potential application? See, for suppose For suppose, so if a person is taster, okay, if a person is a taster, this is 2, right? You don't need 2. Yes. If a person is a taster, okay, if a person is a taster, if a person is a taster, another person is a non-taster. Okay. So in this case, if any one of the child is born with a non-taster, if any one of the child in the F1 generation born with a non-taster, so according to Mendelian laws, 100% offsprings in the F1 generation must be taster. So they produced 4 children. 4 children are tasters. Okay. In another case, first 3 children are taster. 4th children is non-taster. So that means Mendelian laws are saying, Mendelian laws are saying, in F1 generation, 100% tasters. In F1 generation, if any one person is a non-taster, the simple paternity diagnosis. That means first 3 children are got from this male, and the last child is she had affair with another non-taster. That's why non-taster come in the F1 generation. So this is how any violation they use it for the paternity diagnosis, because that rules are 100% accurate. Any deviation is nothing but a mismatched parent. Understand? So now you can see in F2 generation, only 25% of children should born as a non-tasters. If 75% of children are non-tasters, okay, 25% children are taster. This is also deviation. So any deviation of this frequencies, 3:1 ratio of phenotypic ratio, 2:1 genotypic ratio, any deviation is considered as a mismatched parent or paternity diagnosis. Paternity diagnosis. So these are all the frequencies are 100% accurate. If any violation, that couple's mating has a problem. There might be a 3rd person involved or 4th person involved through which they got the children. So that's why these Mendelian laws are used in multiple places in everyday life, in everyday conditions, in everyday practical problems. So one of the topic is like paternity diagnosis that comes into the 12th chapter, that also depend upon the Mendelian laws. So 12th chapter. Understand? So now the 4th law of the Mendel. Now the 4th law of the Mendel is law of independent assortment. Law of independent assortment. So what do you mean by independent assortment? So gametes, you know about, one is taster. Okay, taster, heterozygous taster. So chromosome 1, chromosome 2, chromosome 3. So this is 1, 2. Here somewhere as heterozygous taster, capital T, small t. Okay, here heterozygous roller, capital R, small r. Okay, so here gametes formation. So gametes formation, that is meiosis process. So in gametes formation, so if two gametes are coming, so capital can be with capital R. Okay, so that means if first chromosome comes here, can we say first chromosome of this pair only come in the first gamete? It's not possible. Okay, so it can be paired with capital R or it can be paired with small r. So if first one is paired with capital T is paired with capital R. So then what would happen? So this one and this one is come to this chromosome. So this one and this two obviously pair it. Okay. So or else you can say capital T is paired with small r. Why? Because it's not the fixed. So while gametes formation time, each character behave independently. So that means capital taster gene must be paired with the roller gene. That kind of condition not possible. Taster gene can be paired with roller gene or can be paired with non-roller gene. So it is the independent. What happens, how it happens, nobody predicted. That means it has the probability of pairing with any kind of gene, either capital R, roller gene or non-roller gene. So if it is paired with non-roller gene, then what would happen? Capital T, small r. So capital T, small t, capital R. This is independent assortment. So that means these characters and these characters are independent. So dependent means this is the diagram. So in the same chromosome, capital T, small t is there. Capital R is small r is there in same chromosome. So if one chromosome goes to the one gamete, another chromosome go to the another gamete, then what happens? Capital T, capital R, small t, small r. How many gametes are possible? Only two. Because of independent assortment, number of gametes are 4. Understand? So increasing the number of gametes is due to independent assortment. So you can take third one, diabetes gene. Okay, capital D, small d, diabetic, non-diabetic. So now these 3 character gametes should come. Okay, so now it has the choice of the taster gene. Capital T has the choice of capital R, small r. Okay, so now small t has the choice of, small t also has the choice of capital T, small r. So now these two got over. So now if capital T, capital R, okay, so capital D, that is one occasion. Capital T, capital R, small d, one occasion. Okay, so now you can take capital T, small r, capital D, capital T, small r, small d. So how many? 3. Now you can see. So this is the gamete like capital T, R, D. Okay. So T, capital R, small d. Okay. T, small r, capital D. T, small r, small d. Now small t. Small t, capital R, capital D. Small t, capital R, small d. Small t, small r, capital D. Small t, small r, small d. See how many formed? 1, 2, 3, 4, 5, 6, 7, 8. 2 power 3, 8. 8 genotypes. N is 3 here, 2 power 8, 2 power n, 2 power 3 is equal to 8. So if there is no independent assortment, then what would happen? So if capital T must pair with capital R, okay, capital T must pair with capital R, must pair with capital D, then how many genotypes are possible? TRD is 1, small r is 1. Understand? If it is random, okay, gametes formation, or it is fixed gametes formation. So in our meiosis process, because of crossing over, a chromosome might be exchanging the genes. So if the case is before exchange or in that crossing over process, that character genes are involved or not involved. On the basis we will go. This is how independent assortment. So in the inheritance process, every character behaves as independent of the other. Okay, a taster gene must have inherit together as a roller gene. So these kind of things are there. Tasting gene is an independent gene. It can behave independently. It can pair with the roller gene, non-roller gene. It can pair with the diabetic gene, non-diabetic gene. When it is possible, because the tasting genes are independent. If they are dependent, tasting gene, roller gene, non-diabetic gene together to be inherited, this kind of dependence is there. Number of gametes would reduce, diversity would reduce. Because of these independent assortment only, 16 offsprings come into being, 9 genotypes are coming to being. So this is the law of independent assortment. While formation of the gametes, when more than 2 characters or more than 1 character is involved, so each character, while formation of the gametes, act as a independent rather than depend upon the other character. So that is the independent assortment. That is the last law of Mendelian. So last law of Mendelian. So this is all about today's class. Tomorrow we'll discuss about. So this we call as a multifactorial inheritance. So third, fourth, fifth, this is how we can go multi-factor inheritance. So inheritance we have seen like single factor inheritance. Single factor inheritance through tasting ability. So di-factor inheritance. Taster, tasting and rolling abilities. Okay, trifactor. Tasting ability. Okay, rolling ability, diabetic. This tri-character. So fourth, fifth, and finally multi-factor. So multi-factor inheritance. So in multi-factor inheritance, factors are n. So number of gametes, number of phenotypes, number of offsprings. So these are all the formula I had given. Okay. So this is how n we can substitute. Multi-factor means we have to write this final formula. If n characteristics are involved, what would be the number of gametes, phenotypes, genotypes, offsprings, that you have to write as a formula. So this is all about the Mendelian laws. Law of unity factor, law of segregation, law of dominance and recessive, and finally fourth law of independent assortment. So independent assortment is coming to being in the discussion when 2 or more characters are coming to being. Prior to that, we didn't require the independent assortment inheritance, that law applicability. So in next class, we will see about the human examples, blood group inheritance, sex inheritance, okay, organ transplantation, so paternity diagnosis, missing person diagnosis. There are so many areas Mendel laws can be applied in the human. As the same well, there are so many areas these Mendel laws are not applicable in the human beings. These are all the potential questions already they gave in the exam. So if you have any doubts, you can let me know. So if you have any doubts, you can let me know, or else we will discuss. So tomorrow, is it possible for you? Tomorrow, do you have any separate plans for Sunday? Yes, Praniti, are you there or not? Okay, we will meet on Monday. Just you can revise this one. Okay, you can. So these are all basics. Nobody will ask you in the exam. So without understanding these questions, you could not understand the potential questions coming in the exam. These are all the basics. You don't need to worry. Still, you want too much of knowledge from these classes, you can watch one more time. If you have any doubts, you can ask me. I will explain it.